Hello, biologists. So in making this video, I am looking to put together something that's both comprehensive and informative for you when you're reviewing for your test. Because oftentimes with this unit, I feel like students ask for a lot of extra help, but they're sort of asking the same questions. So I want to go from start to finish through aerobic cell respiration and photosynthesis, those being the most complex of the processes we've talked about in this unit. From start to finish for both, so I apologize for the length of the video. But without further ado, we're going to start with cell respiration and then get into photosynthesis. So I guess first and foremost is worth noting, okay, when do we do this thing called cell respiration? Particularly of the aerobic variety. Now, say our friend here is going out for a run. Okay? If she were to engage in an all-out sprint for something under than 2 minutes, her muscles are going to go through what's called lactic acid fermentation, which hopefully you know something about. However, if she decides to elongate her run, and blood is actually able to travel to that muscle that's constantly in need of ATP, then we are going to do this thing called aerobic cell respiration. That being said, that process of aerobic cell respiration is much different from respiration as a process of gas exchange. So when we think of respiration, we should think of the inhalation of oxygen and the exhalation of carbon dioxide. Whereas cellular respiration is the exergonic process whereby we break down glucose, okay, to produce ATP. And so if we take a look at our overall equation, okay, we'll see glucose and oxygen are our main inputs in this net overall equation. Okay, carbon dioxide, water, ATP, and heat. However, we know that it's important for us to understand aerobic cell respiration as a series of redox reactions. That meaning, like any other ship name, when we see reduction, we see oxidation. And accordingly, we can see here in this slide that when glucose is oxidized, that means it's losing hydrogen atoms, but more importantly, it's losing electrons. Remember, Leo the lion says Gerd. Okay, so losing electrons is oxidation, and subsequently, oxygen is going to be reduced to form water as our sort of last stage of cellular respiration. Of course, the goal of cell respiration is to harvest the energy stored in the chemical potential energy stored in the bonds of glucose, okay, and to convert that into something more usable for the cell, like ATP. I think there was an analogy in a video that I shared with you guys that said that if you had a summer job where you were working in an ice cream shop and your manager decided to pay you in ice cream cones rather than in money, well, how useful would that pay be? So in a similar scenario, we have to think about ATP as sort of this energy currency for our cell. So, accordingly, one of the other more important players in cell respiration is this thing called NAD plus and NADH. NAD plus, this positively charged molecule here, is we'll see it's commonly reduced. And remember, gaining electrons is reduction. So, in doing so, NADH is one of our most important players because of its role as an electron carrier. Okay, we'll see numerous instances where NAD plus gets reduced to form NADH all throughout. So just looking through sort of the process as a whole and how photosynthesis and cell respiration play together with one another. Okay? If we think about just the production of cell respiration and photosynthesis, okay, Photosynthesis is going to produce for us O2, which we need to do this thing called aerobic cell respiration. Aerobic cell respiration is going to produce for us CO2 and H2O, the thing we need to do photosynthesis, and the cycle sort of propagates itself. Okay? But zooming back into aerobic cell respiration, let's take a look first at where exactly this is taking place. Okay? So, glycolysis, one would think, because knowing that, quote-unquote, the mitochondria is the powerhouse of the cell, one would think that, okay, glycolysis, like all other stages of aerobic cell respiration, take place in the mitochondria, but that's false. Okay? The glycolysis reactions are going to take place in the cytoplasm, or cytosol as your textbook lists it. Okay? The rest of these processes, pyruvate oxidation, citric acid cycle, oxidative phosphorylation, they're all going to take place in the mitochondria. Okay? Our citric acid cycle, those important enzymes are going to be housed in the matrix of the mitochondria. And finally, oxidative phosphorylation, we know, is going to take place somewhere in the inner membrane of our mitochondria, but more on that later. Okay? I want you to also take note of the different kinds of phosphorylation that take place throughout aerobic cell respiration. So keep in mind that, unlike oxidative phosphorylation, the processes of citric acid cycle and glycolysis are going to entail substrate level phosphorylation as opposed to oxidative phosphorylation, a detail in which I won't go too in depth in this video. Keep in mind, just so you can recall, that phosphorylation just refers to the addition of a phosphate group to any particular molecule. Okay? In this case, we're talking about phosphorylation as it applies to phosphorylating our ADP molecule, adenosine diphosphate, to form ATP. Because after all, our goal is to make this thing called ATP. That being said, not all of the chemical potential energy from glucose can actually be used to make ATP. In fact, 2/3 of this energy is going to be released as heat, one of cellular respiration's byproducts. Okay? So, moving swiftly along, right into glycolysis. Okay? What you see on your screen now is our net equation for glycolysis. So what's going in, what's our quote-unquote input, is glucose, one glucose molecule. Note that we have 1, 2, 3, 4, 5, 6 of these gray beads, signifying each of them a carbon atom. Okay? And through glycolysis, we are going to produce 2-pyruvate, this 1, 2, 3 carbon sugar. So, note that in terms of the conservation of mass, no carbon have been lost here. Okay? But something seems to be going on in terms of reduction and phosphorylation throughout this process. Okay? So, now, the substrate level phosphorylation that takes place, as you'll see in the process of glycolysis, okay, happens like you'll see on your screen right now. Okay? So this is one of our intermediates that will form later on in glycolysis. But if you take a look at sort of our before and after here of this enzyme catalyzed reaction, we'll see that this phosphate group from our intermediate is being cleaved off. Okay? Note the difference between substrate and products is a loss of a phosphate group. However, the change being is that this ADP molecule has been phosphorylated to form ATP. And so what happens is this enzyme is going to squeeze together the phosphate from that intermediate with our ADP molecule to form ATP. And so if we look at the steps of glycolysis, okay, you'll see that, okay, first of all, we have our energy investment phase. Okay? So what that means is, and this is the analogy I've used in class before, why would we invest in the stock market? Well, we'd invest in the stock market to try to make money. How do we invest in the stock market? Well, we have to spend some money in order to make that money. And so, just like that analogy, we have to spend ATP in order to, in the long run, make some sort of substantial ATP. Okay? So you'll see here in steps 1 and 3, okay, that we are spending 1 molecule of ATP. So that is 2 molecules of ATP that we are spending. Okay? And note that this phosphate group from our ATP is being tacked on to our intermediates here. Okay? So note that the difference in step 1 between glucose and glucose 6-phosphate is just the addition of this P here. And the difference between fructose 6-phosphate and fructose 1,6-biphosphate is these 2 phosphates here at the end. Okay? And so as sort of the last step here, we have the splitting of our 6-carbon chain into 2 phosphorylated 3-carbon chains. So, moving on, okay, our first step of our energy payoff phase, which is happening not once but twice because we've split our 6-carbon chain into 2. Okay? Our first step is to phosphorylate this molecule, but in doing so, that entails the reduction of NAD plus to form NADH. Okay? Note that that happens on both sides. Okay? They're identical molecules and identical reactions. Okay? And moving forward, we have the production of 1, 2, 3, 4 ATP, each of which have been formed from using the phosphate group to tack onto ADP to form ATP, our classic example of substrate-level phosphorylation. Okay? And we see that down here with phosphenylpyruvate losing that final phosphate group to form pyruvate, a 3-carbon sugar. So those are 2 classic examples of substrate-level phosphorylation. Okay? Next, what's going to happen is that pyruvate is going to be converted into this thing called acetyl coenzyme A in a step called pyruvate oxidation. So what exactly is going on here? Well, if we sort of just look at our carbon molecules here, we'll note that pyruvate has 1 more carbon than acetyl coenzyme A. Okay? So where did that carbon go? That carbon was lost as CO2. Okay? What is coenzyme A? I've said this many times. It's like an Uber or limo or a taxi cab driver that is going to bring this 2-carbon molecule, which we have a fancy name for. We call it an acetyl group, to the citric acid cycle. Now, because we are oxidizing pyruvate, we know redox. We always see oxidation and reduction together. Where is the reduction, you may ask? The reduction is the NAD plus being reduced to form NADH, our favorite electron carrier. Okay? So next, okay? And keep in mind, just an important note, that this is happening, as you see here, twice per molecule of glucose. Keep in mind that we formed 2 pyruvate, so you really should be multiplying what you see here by 2, because we formed 2 acetyl-CoA, and those 2 acetyl-CoA will happen in parallel Krebs cycles. Okay? So per glucose molecule, this is all happening twice. So if we just zoom out here and we think about the citric acid cycle, then we're looking at this acetyl coenzyme A molecule. Well, immediately this limo driver is going to leave. That limo driver is done for the rest of time. He's going to be recycled to be used in pyruvate oxidation again. Okay? going through the citric acid cycle, we are producing CO2, we are losing more carbon, okay? We're producing 3 NADH, 1 ATP, and 1 FADH. And if you're doing the math at home, which isn't hard, really this is 4 CO2, 6 NADH, 2 ATP, and 2 FADH2. Okay? If we look at the more complex version of all of this Krebs cycle nonsense, then, okay, this 2-carbon acetyl group is going to enter our citric acid cycle. Okay? That's immediately going to combine with a 4-carbon molecule by the name of oxaloacetate to form citrate, for which the citric acid cycle is named. Now, we're going to form another number of intermediates, alpha-ketoglutarate, succinate, fumarate, malate, and then back we return to oxaloacetate. So we regenerate the thing with which we started. So it's noted that this is sort of an end for our carbon molecules. Okay, this is the end for all of this. So what then is going to go into our next step, oxidative phosphorylation? Well, we've been talking about the production of NADH and FADH2 this whole time. And if you've been doing the math, we have 10 NADH and 2 FADH2, which are going to go into the electron transport chain. Now, some of you may ask, what's the difference between NADH and FADH2? Okay, NADH and FADH2 have identical roles in that they are electron carriers. They are going to bring electrons to our electron transport chain. Okay? The difference being is that FADH2, as you can see in our diagram here, and as you can see on the slide on your screen, okay, FADH2 enters the electron transport chain at a slightly later spot than NADH. The result is that per FADH2 molecule, we'll get less ATP than we would from NADH. That being said, okay, let's talk about oxidative phosphorylation. So, the term itself, okay, oxidative phosphorylation. So, let's start with the beginning. Oxidative. So, that has something to do with oxidation. Okay, if we look here at NADH and FADH2, we should note that they are both losing electrons. Remember, losing electrons is oxidation. That is the start of our process. Okay? And the end result being the production of ATP from ADP plus P. Okay? So we start with oxidation and we finish with phosphorylation. Okay? The whole process gets that name because it's named for both the beginning and the end. Now, oxidative phosphorylation consists of 2 components, the electron transport chain and chemiosmosis, which is a special term for the diffusion of chemicals. Now, what's going on in the electron transport chain? Well, NADH is going to drop its electrons off at these membrane-bound proteins that are found in the inner mitochondrial membrane. Okay? We know this is a membrane by the presence of these phospholipid molecules here. Anyway, the electrons are dropped off at these particular embedded proteins, and in moving from each protein to each protein to each protein, they're emitting a little bit of energy. Okay? What is that energy used to do? Well, if you can recall from our last section, we require energy to move against concentration gradients. That is, to actively transport something. So what are we actively transporting? We are actively transporting H plus ions, okay? And we call those protons because hydrogen has 1 proton and 1 electron if it is neutral. And if we take away that electron, then it's just a proton. So it is used to actively transport H plus into this thing called the intermembrane space. That is between the outer membrane and the inner membrane. So we have this accumulation of H+ ions all throughout our intermembrane space. Now, what is the fate of them? Okay, these H+ ions are going to be used to flow through ATP synthase. Okay, and that process is called chemiosmosis. So the H+ ions flow through ATP synthase. That is like their ATP synthase's fuel. Okay, ATP synthase is a motor protein that we'll see again in the light dependent reaction of photosynthesis. But the H+ ions are going to flow through a channel that you can see on your screen now in that molecule. Okay, that's going to turn the top of that molecule, and that is going to cause ADP plus P to squeeze together to form ATP. Okay, and that will be done in the mitochondrial matrix as we've seen here. As a result, we form about 28 ATP from this process on average, which gives us a grand total of 32 ATP from aerobic cell respiration. Compare that to any kind of fermentation from which you only get 2 ATP per turn, and this is a much higher energy yielding process. Okay? However, it is going to take longer, which in the case of a sprint does not seem as time efficient. Okay? So, moving on. Going from cell respiration to photosynthesis now. Okay? And so photosynthesis is quite the opposite. Photosynthesis is literally, if we take the last stage, so we're going from breaking glucose, oxidizing glucose to form ATP, and in photosynthesis we're actually using ATP and NADPH to help make glucose. So we're more or less undoing the thing that we just talked about doing. Okay? And so it's all based on our ability to use light energy. I'm going to go into this a bit later, so I'm not going to talk about it quite yet. So just some basics. Where does photosynthesis occur? Okay? Well, if we cut open a leaf, if we cut a leaf in half, we look at it under a microscope, we'll see this mesophyll section here. This mesophyll section is going to be full of these chloroplast-rich cells, okay, as denoted by these green circles throughout. Okay? Note that here at the bottom of our leaf, we have tiny pores called stoma or stomata that allow CO2 and O2 to travel in and out respectively. Now, taking a look zooming a little closer here, okay, we take a look at, okay, where does this actually happen in the chloroplast? Well, we have two different sections, okay? And accordingly, we have two different reactions, okay? So our thylakoids are thin discs, okay, which a stack of which are called grana, are going to be the site of our light-dependent reactions, okay? And our stroma, on the other hand, is going to be the home of what are called our dark reactions, light-independent reactions, or the Calvin cycle. Those three terms are all interchangeable in my mind. So, just like our process of aerobic cellular respiration, photosynthesis is also a series of redox reactions. And so displayed on your screen should be the overall equation for photosynthesis. We have CO2 getting reduced and H2O being oxidized to form glucose and oxygen respectively. But that doesn't tell us a whole lot, because if you've been in class, you know that the mechanism by which is quite complicated. And so, if we're looking at this, okay, now let's finally take a look at our two sets of reactions here. So we have light coming in, okay? H2O being used in our light reaction to produce ATP and NADPH. Oxygen is produced as a waste. And these two molecules are going to be used to help convert some molecule into glucose in the Calvin cycle. Again, take note, light reactions as symbolized by these disks are taking place in the thylakoid. The Calvin cycle is taking place in that thick, syrupy fluid in the stroma. Now, ATP and NADPH are not the only inputs into this process, but CO2 is also going to be used. Okay? And that's significant for a particular reason that I'll get to in a few minutes here. So, photosynthesis all revolves around our ability to use light energy to convert that into chemical energy. And if you remember my little lighting a stick on fire and showing you that it turns a different color, okay? When we excite electrons, those electrons are going to be in an excited state. That excited state doesn't last for long. In that electron coming back down to what's called the ground state, its original level, okay, some energy, according to the law of conservation of energy and mass, some energy is going to be conserved, all energy is going to be conserved, and it's going to be transferred as heat and, some sort of photon, some fluorescence. Now, why this is important? Well, we need to get into our light-dependent reaction, which consists of, as you see on your screen, two of these photosystems here. Okay? So, what is a photosystem, you may ask? Okay, so a photosystem is an embedded protein that consists of light harvesting complex. I've compared them to antennae that gather light. Okay? It consists of a reaction center complex and a primary electron acceptor. So, here's what's going to happen. Okay? And note that there are also these chlorophyll A molecules, which I seem to have neglected. But those are going to be very important, and they are a crucial pigment molecule for plants. Okay? And so light is going to enter our photosystem. And our light-dependent reaction starts with photosystem 2. Oops, if you'll excuse me, I need to grab my charger. As I was saying, our light-dependent reaction begins with photosystem 2. Okay? Now, that may make no sense. If you recall in class, that's because scientists discovered photosystem 2 second, so it got sort of the short end of the stick. Okay? So light is going to strike the light harvesting complexes of photosystem 2. Okay? Those molecules together are going to vibrate and hit one another until they transfer that energy to our chlorophyll A molecule. That's going to cause the excitation of some electron. That electron is going to travel to a primary electron acceptor. Then that electron will travel again down yet another electron transport chain, which, in case you forgot, was a series of proteins embedded in some membrane. In this case, it's the membrane of the thylakoid, okay, that are going to be used to harvest energy from high-energy electrons. And so, eventually, this electron is going to reach photosystem 1. Okay? In photosystem 1, same thing happens here. Light is going to strike the light harvesting complexes. Those molecules are going to vibrate. Okay? That's going to excite an electron in chlorophyll A. That electron is going to be excited. This time, we are going to use that energy to produce NADPH. Okay? That NADPH will be a major input for our Calvin cycle. Now, it should be noted that, okay, what did we use the energy from photosystem 2 to do then? Well, if I reveal everything here, okay, in our thylakoid space, the energy from our first excited electron has been used to actively transport H plus into the thylakoid space. The question is, okay, where did those H plus come from? Well, in order to replenish electrons that have been excited, okay, water is split, okay, into its component parts, into oxygen and hydrogen ions. Okay? In doing so, we've produced a bunch of these H plus ions. We also have in the stroma an accumulation of H plus ions as well. Okay? We'll see where those come from later. So, since we have now a high concentration of these H plus ions, okay, in our thylakoid space, those can again be used to power ATP synthase. And of course we know ATP synthase's role is to synthesize ATP from ADP through the power of chemiosmosis, H plus ions flowing through that particular membrane protein. Okay? And now we're off to the Calvin cycle. So, first step of our Calvin cycle is, okay, we need CO2. Plants breathe in CO2. So we are going to combine CO2, and you'll see on your screen that these numbers here, okay, the 3 and the 6 in front of 3 PGA here, and the 3 in front of RUBP, consist of the number of those molecules involved in the process. Okay? So in step 1, we're going to take 3 CO2 molecules and 3 RUBP molecules and form 6 3-PGA molecules. Okay, well, that doesn't sound very important. However, it's important to note that this is where atmospheric carbon, that is carbon dioxide, now becomes usable for the biosphere, living things. Okay? So, to recap, CO2 combines with RUBP. The enzyme responsible is called Rubisco, and we form this 6 3-PGA molecule. Reduction. Okay, so these 6 3-PGA molecules are going to be reduced. In doing so, okay, we are going to use the electron carriers and ATP to make that happen. So here we have the step by which we're taking our chemical energy, our electron carrier and our ATP, and we're putting it into our G3P molecule. So the goal here was really to build G3P, which is going to then come out of our cycle to form glucose or some other sugar. Okay? And then, of course, rounding out, what's going to happen is that this G3P is going to reform RUBP, and that is the Calvin cycle. Okay? So, on my end, I think that's it. If you're watching this video, good luck on your test. Make sure to subscribe and hit the like button. Hopefully we can get over 2 likes. And you keep on keeping on.